Progress.
Will, can you share your screen, please?
Yes. Does that look correct on your end?
It looks it's starting. That's good.
Yes.
Okay, so the last talk of the day is from Will Conway from the New York Structural Biology Center, and he's going to talk about the assembly of the mitotic spindle. Completely different than the talks of the rest of the day, at a different level. So we look forward to this. Thank you very much, Will.
Hi, thanks so much for the introduction and the invitation. I'm going to talk today about our work that we've been doing trying to visualize structures in the mitotic spindle in human cells. And as you alluded to, this is certainly a slightly different theme than the earlier talks, but I think many of the principles are still fundamentally the same. Sorry. And I think that sort of gets at one of the core thesis and hypothesis of structural biology, which is that visualizing complex and higher order structures can tell us the rules of their organization. And I think this is true across a variety of length scales. Visualizing protein structures gave us great insights into how atoms organize first into sequences of amino acids, and then to secondary structures that fold into more complex geometries that allow enzymatic activity. Similarly, at the scale of individual cells organizing into tissue layers, complexity arises from collective organizations between individual components that let you do things that you're not able to do with individual cells. And even up at the overall much higher length scales of organisms interacting in ecosystems, flocks of sheep start looking a lot like fluids as they move from one pasture into the other. And so understanding the rules of how these systems organize and are able to do their function really is at their core a structural question. In terms of the spindle, what I'm really interested in understanding is how interactions between individual microtubule filaments and molecular motors can assemble mesoscale bundle-like structures that are then critical to the overall function of the spindle itself. And I think this is a broad question that's true in a lot of cellular organelles and has sort of become a defining struggle in a lot of biophysical modeling, is how we can think about these questions that bridge length scales. To give you an introduction to the overall biology of the spindle is the organelle that's responsible for segregating chromosomes during eukaryotic cell division. On the left, I'm showing a cartoon of a spindle in metaphase, where the chromosomes are all oriented on the metaphase plate, and they're bioriented, where microtubule filaments in the spindle are connecting them to opposite spindle poles, which are formed by centrosomes. And this biorientation is crucial because it ensures that one copy of each chromosome ends up in each daughter cell after cell division. Microtubules are polar filaments. They polymerize at their minus ends and grow rapidly towards their plus ends. At some intrinsic rate, they undergo what's called a catastrophe, where they switch from polymerizing to depolymerizing, and then they shrink all the way back to their minus end where they vanish. In principle, they can also undergo what's called a rescue and switch from depolymerizing back to polymerizing, but the rescue rates in the spindle are so low and they're typically neglected. Microtubules can interact with each other via molecular motors, which crosslink and slide anti-parallel microtubules, which generate forces and connections that are essential for assembling the spindle. A balance of two motors are critical to ensuring proper assembly of the human metaphase mitotic spindle. This is EG5, which is an anti-parallel crosslinker that walks towards microtubule plus ends and slides them. And EG5 is balanced by dynein, which is a slightly different structural motor. It walks along microtubules towards the minus ends and transports an adjacent microtubule like cargo, thereby clustering the microtubule minus ends. Inhibiting either motor has really dramatic consequences for the overall geometric organization of the spindle. Inhibiting EG5 gives you an aster-like monopole, which I'm showing on the top of this little diagram on the right. Inhibiting dynein defocuses the spindle poles and gives you this disordered wispy structure. And remarkably, inhibiting both of the motors actually gets you back to something that looks a lot like a bipolar spindle. And things that we really have struggled to understand in the field is why are these motors seemingly interchangeable, even though they have very different mechanisms of action, and why does inhibiting both restore you back to the same equilibrium? The classical textbook picture of how all this is structurally organized is that all of the microtubules nucleate at the spindle poles and grow inwards to search for and capture chromosomes. Most of this theory is based on in vitro reconstructions of microtubules near centrosomes, where we can see that they nucleate and grow radially out, and light microscopy of spindles, where we can see with diffraction limit and resolution that there's continuous tubulin staining in green between the chromosomes in red and the poles. Unfortunately, at the diffraction limit of light microscopy, we really don't have the resolution to explicitly test whether or not these predictions are true, because we can't observe individual microtubules in these dense bundles. And I think this gets at what's sort of the core problem with trying to think about a lot of these molecular to cellular questions, that we have this really frustrating experimental gap where it's very technically challenging to observe individual molecules interacting in a cellular environment. With light microscopy, we can see the entire cell and develop continuum models at the length scale of microns and the time scale of seconds. And using advances in cryo-electron microscopy that we've heard about earlier today, we can really start to push down towards angstrom resolution of big macromolecular complexes. And with molecular dynamics, we can simulate their motion at the order of microseconds, but this requires reconstituting the systems outside of the cellular environment. So we don't learn very much about how these components are actually interacting inside of cells. So we're missing this core intermediate resolution technology that will enable us to connect angstroms to microns over a length scale of 4 orders of magnitude of difference in scale. The solution lies in recent advances in cellular electron tomography. The core challenge that has to be overcome here is that samples more than 200 to 300 nanometers are very electron opaque. So we somehow need to thin the sample in order to put it into an electron microscope and observe these structures beyond the diffraction limit of light. There's two workarounds that have been developed in the field. One is to use serial section plastic electron tomography, where we embed the entire cell into a resin, and then we cut thin sections with what's called a cryo ultramicrotome. And this allows us to pull the sections out in the ribbon, and we can acquire consecutive sections in serial to stitch the entire structure back together. While this gives us the full structure, unfortunately, due to heavy metal staining that we need to enhance contrast in the plastic, we're stuck at a maximum resolution of about 5 nanometers because the heavy metal stains are just physically that big. To push all the way down to near atomic resolution, we can use cryo-electron tomography. The trade-off here is that we need to mill thin sections into cells, like this cartoon I'm showing on the top right, where we've used a focused ion beam and a process called fib milling to physically ablate material on either side of a very thin section. In a typical fib milling experiment, we destroy 99% of the cellular material. So we're only looking at a very small subsection of our overall structure. However, we're able to get much higher resolution imaging, so we can visualize the interactions of individual microtubules and the positions of molecular motors. So using a combination of these two technologies, we can see the entire structure and then get detailed information about the interactions of microtubules and motors. When I was in graduate school, we did a lot of this serial section imaging. We cut thin slices, and how tomography works is we take tilt series of these slices, and then using the Fourier slice theorem, we can reconstruct entire volumes. Because this is all in plastic, we can take serial sections and begin to stitch entire cells back together. And what this let us do was to solve what we would call the first structures of the spindle. We solved the first worm spindle in 2017 and the first human spindle in 2022. We're actually able to observe the trajectories of every microtubule in a spindle inside of a human cell. And the results are completely inconsistent with what classic textbook pictures tell us should be happening. Only about 20% of the microtubules nucleate at either of the two spindle poles, which I'm showing here on the left, and very, very few of these microtubules are long enough to interact with a microtubule that nucleated at the opposite spindle pole. The remaining 60% of the microtubules are nucleating somewhere else in the spindle bulk. Because of that, we're not able to assign the polarity directly from the plastic imaging. And what the function of these microtubules are really isn't super clear. So we need to somehow be able to visualize interactions with motors and the polarity of these microtubules to be able to get a consistent picture of how the spindle assembles. Our first try was to avoid doing any cryoimaging because it seemed very challenging and like a lot of work. So we tried to just infer what the polarity of the microtubules were and compare to bulk fluorescence measurements to verify our inference. We said, okay, probably every microtubule somehow nucleating either from branching or from some sort of transport from the pole that is closest to. So we can just assign each microtubule to the closest pole, and this will give us polarity inference. When we compare to bulk measurements of microtubule polarity from second harmonic imaging, we see that the agreement between these two really isn't very good. Our predictions from this branching nucleation model predict that the spindle should be almost completely parallel on either side of the metaphase plate. And what we see, in fact, is that it's much closer to 50-50 polarity. So really, we do need to actually do cryo-electron microscopy imaging to visualize the microtubule polarity directly. So 3 years later, we've been able to achieve this. And I'm definitely skipping over a decent amount of the sample prep steps here. But the basic procedure here is that, again, we mill thin sections into intact vitreous cells, and then we use new advancements in processing technology in subtomogram averaging that allow us to crop small subtomograms along each microtubule and average to get an overall structure. This tells us the polarity of the microtubules from the tubulin slew that you can see as this clock-like gear around the edge of the microtubule on each tubulin. On the right, I'm showing a sample tomogram that we acquired in a metaphase spindle where I've colored the microtubules blue and orange based on the relative polarity. I'm also showing where the ribosomes are in gray, and I think this really emphasizes that the cellular environment is just so crowded, and all of these interactions are just incredibly complex and happening in dense environments. The other thing that really jumps out is that we see a lot of anti-parallel interactions within each of these microtubule bundles. And as we did some more quantification to try and think about this a little more carefully, we see that within each of our tomograms, we observe what appear to be quite dense regions that are bundles of microtubules. Within these bundles, we see consistent spacing between adjacent microtubules. For center to center, that spacing is about 33 nanometers. You can see that here in this histogram on the top right. We're plotting the frequency of spacing between microtubules, and there's a very, very sharp peak at 33 nanometers. We also quantified the relative polarity as a function of distance between the microtubules. And for nearest neighbors, the microtubules are anti-parallel with their neighbors about 40% of the time, which isn't consistent with this traditional picture where all the microtubules nucleate at the spindle poles. What we're actually seeing is that the spindle is this rich, interdigitated structure where there's anti-parallel interactions all over the place that can be generating forces. So while this gives us some initial structural information, it really raises a lot more questions than answers about how any of this can really be working. One thing that's really striking is that we don't see any difference in spacing between parallel and anti-parallel microtubules. We know that different motors are all different sizes. For example, egg5 is about 80 nanometers large and Dynein is about 55 nanometers tall, and have different parallel and anti-parallel preferences, but we don't seem to see any difference between parallel and anti-parallel microtubules. that will be consistent with different motors organizing these bundles with different polarity preferences. It's also not super clear where these anti-parallel microtubules that we observed can be coming from. One possibility is that we have really, really extensive branching nucleation from the opposite pole, which would allow you to keep building over until you reach the other side. Alternatively, these structures really could be locally organized in a bottom-up manner by molecular motors to generate anti-parallel structures where you could generate force. To try and answer these questions, we did a perturbation experiment where we inhibited duplication of the centrosomes. And 48 hours later, after 2 rounds of division, we've gone from 2 mother-daughter pairs to a single mother centriole in each spindle. And as a result, the spindles spontaneously form these motor-active monopolar geometries where all of the chromosomes are on the outside surrounding a single pole. This allows us to test whether the anti-parallel interactions that we observe are from branching nucleation coming from the other side or locally generated because we physically remove the other pole that could potentially be the source. What we see ends up being very striking. If you ask the question, is spindle bipolarity required to form these anti-parallel interactions? The answer seems to be a very clear no. I'm showing on the left a sample reconstruction of a microtubule bundle in one of these monopolar spindles. And again, you can see very clearly there's a lot of anti-parallel interactions that are forming in the monopolar case where it's not possible that the microtubules branch from the opposite pole. If we do more careful quantification, we see that the spacing and polarity in monopolar and wild-type metaphase spindles are very, very similar. Taken together, these results indicate the spindle really is locally organized by interactions that are happening between microtubules at the length scale of microtubules themselves, and not by branching from the poles, which is to say the spindle really is organized bottom up, not top down as predicted in traditional models. This, of course, again, raises more questions. That if it isn't the spindle geometry that's organizing the microtubules, it must somehow be the molecular motors, but how exactly is all of this happening? Again, the 2 key motors here are EG5 and dynein, and perturbing either of them gives dramatic reorganization of the spindle geometry. We first want to see if we could figure out where this 33 nanometer spacing between the microtubules came from. So we inhibited both of the motors, again did reconstruction, and looked at the spacing between the microtubules. We see that inhibiting dynein dramatically increases the spacing between microtubules from about 33 to 40 nanometers from center to center. However, inhibiting both of the motors restores the wild-type spacing. This failed epistasis test indicates that dynein can't directly be cross-linking the microtubules because if that were true, the spacing would remain elevated in the dual knockout case. It suggests there's some other, likely apolar process that's organizing the microtubules. It's also noteworthy that the spacing is very tight. The spacing between adjacent microtubules, if you look at the wall-to-wall between the filaments, is only 8 nanometers, which really isn't a lot of space for motors to be able to fit. We hypothesized that perhaps steric interactions between the microtubules are what are responsible. And if this were true, the microtubule spacing should scale with density. So we went back and looked at what the local density of microtubules was in each of these bundles and plotted density versus spacing for all of our motor knockout conditions. And you start seeing a very clear pattern forming. That inhibiting dynein increases the spacing and decreases the density. Inhibiting EG5 actually very slightly increases the spacing and more dramatically increases the density. Inhibiting both motors sends you back to wild-type density and back to wild-type spacing. So this starts painting a picture where it's density-driven steric interactions between adjacent microtubules that organize bundles, not direct cross-linking by molecular motors. To test this more thoroughly, we went back to the original plastic reconstructions where we can still see microtubule spacing. And we get much better data coverage across the entire spindle than we do in thin cryo-lamella, particularly in low-density regions that we don't typically acquire when we do cryo-ET. If all the microtubules were organized by a crosslinker, you would expect for this microtubule spacing to remain the same independent of density. This clearly isn't what we see. In more dense regions of the spindle, the microtubules are spaced more closely together. This indicates that density-driven steric interactions are responsible for organizing microtubule bundles, not direct cross-linked by molecular motors. Nonetheless, the motors are still somehow critically important, and we know this because inhibiting them reorganizes the spindle geometry. We quantified how the polarity of adjacent microtubules changes when the motors are inhibited, And we see that inhibiting the EG5 motor dramatically increases the fraction of microtubules that are parallel in both monopolar and bipolar spindles. So if we compare our motor active monopoles to EG5 inhibited monopoles, we see that the parallel fraction is much higher. Similarly, if we compare bipolar spindles that are formed either by inhibiting both motors, so EG5 is still inhibited, or in wild-type metaphase, we see the fraction of microtubules that are parallel with their nearest neighbors are dramatically higher. Taken together, this brings us to a novel view of spindle assembly, where the data that we see in the cryolamella isn't consistent with classic top-down pole-centric models, where long microtubules nucleate into the pole and grow inwards, or with a short fix, where short microtubules are nucleating into the pole and are growing inwards by successive branching nucleation interactions. Instead, our data support a bottom-up self-organized model, where microtubules are locally organizing due to steric interactions and interactions with molecular motors. In this view, a balance of the EG5 and dynein motors regulate microtubule density. High EG5 means the density is low because EG5 has extensile activity. High dynein drives high density as it clusters microtubule minus ends and brings things together. And we get an optimal density structure and therefore spindle formation when the two motors are balanced. Steric interactions between these dense microtubules set the rules of spacing between adjacent microtubules with no need to invoke direct cross-linking by molecular motors. And then a balance of forces within these bundles between EG5 and dynein sort microtubule polarity and are responsible for maintaining the bipolar spindle. If you're carefully doing math, you do see there's a sort of key geometric problem here, which is that in these microtubule bundles, there's only 8 nanometer wall-to-wall spacing between the microtubules, but both EG5 and dynein are too large to fit in those gaps. Dynein, for example, is a 55 nanometer tall motor that isn't able to fit in these small gaps that are generated by density-driven steric interactions in the microtubules. And I think this raises two sort of core questions of organization that are closely related. One, what determines the consensus behavior in these systems with different sized components when we see a single spacing in bundle lattices? And two, how do different size motors navigate the size mismatch to still work cooperatively? There's 3, I think, formally possible ways this could happen. One is you could have lattices that have multiple different spacings. So adjacent microtubules don't necessarily have to have the same spacing. We don't see that in the spindle, but it could certainly be happening in other cytoskeletal systems. Another possibility is that motors could localize on the edge of the bundles. And so this could resemble a coaxial cable, where the motors are on the edge and the microtubules are in the core. The third possibility is motors could localize in lattice defect gaps where a microtubule was missing, and they could have more room to maneuver in the second or third coordination shell of the lattice. We wanted to try and explicitly test what was happening here in a controlled system, so we collaborated with Zvonimir Dojic's group at UC Santa Barbara. They assemble in vitro reconstructions of microtubules and molecular motors that generate these extensile active gels. And the nice part here is that we can directly tune different components to explicitly test models of collective organization in bundles. Here they're using kinesin 1 motors that are assembled in aggregates, so we can tune the size of the motor by tuning the size of the crosslinker between the two motor heads. We tried 2 different motors, one's using a streptavidin linker and one using an apiferidin linker. That gave us a final motor size of 10 or 20 nanometers. In both of these cases, we're able to assemble these active gel systems where you can see the microtubules swirling around in an in vitro reconstituting system. And then we can again do cryo-ET to measure local polarity and spacing of microtubules. We saw that very similar bundles formed whether we use no motors, a small motor, or a large motor in microtubules interacting with a pegdepletin. All these motors had very similar 35 nanometer spacing and had very characteristic hexagonal geometry with their nearest neighbors. And again, if you're carefully doing math, this 35 nanometer spacing is too large for either the small or the large motor to directly fit between adjacent microtubules. Nonetheless, we still see, as we did in the spindle, that there's polarity sorting that happens in these lattices. So if we look at the parallel fraction scales with the spacing between adjacent microtubules, we see that for nearest neighbor microtubules, adding the motors makes you about 10 to 20% more likely to be parallel with your neighbors. So again, we have the same paradox here that the motors don't fit directly between adjacent microtubules, but they're still capable of sorting polarity, and certainly we can see that they're capable of generating motion at these large micron scales. To then test directly where the motors end up, we engineered a novel tag with the apopharidon cluster to allow us to visualize where individual motors were in the bundles. I'm showing some examples of this here between the raw tomogram slice and our reconstructed model. And the apopharidon is the circle that shows up in the center of these four images. And then using this, I can just directly reconstruct where all the motors are in the system, and we can start testing hypotheses of how the motors interact with the microtubules. What we see is that almost all of the motors end up on the edge of the bundles. So what I'm showing here is a 2D projection along the bundle axis of this same system. In the top left, I have circles where all of the microtubules and the lattices are. And then I've attempted to render an excluded volume plot where gray shows the excluded volume formed by the microtubules in these systems, and white shows gaps in the lattices where the motors are physically excluded from getting in from the outside by adjacent microtubules that form a wall that the motor can't cross. Then because from the apopharidon tagging, we know where all the individual motors are, I can just count when they end up in these gaps or in the edges, and we see that 93% of microtubules that are bound, 93% of motors that are bound to a microtubule are localized out on the edges of the bundle, not in the gaps. An interesting thing is that the way these bundles seem to form, and this seems true in the spindle as well, isn't a nice surface tension defined circular object. They form these bizarre fractal geometries where there are these canyons and valleys that allow the motors to interact with many more microtubules than they could if this bundle was densely packed in a circular hexagon. On the right, I'm showing a plot of how many microtubules would be exposed if we had perfect hexagonal packing, or a case where we had perfect fractal packing that allowed every microtubule to interact with motors. And we see that in the systems of motors plus the peg, almost all of the microtubules are somehow able to interact with motors due to this bizarre packing geometry. Comparing to a peg alone control where there's no motors indicates this really is a property of the packing itself, not some feature of the motors inducing instability in the bundles. There's two, I think, cool consequences of this. One is that you end up with these excluded cores where the motors can't interact with microtubules anymore. And we see that in these excluded cores where microtubules would have 6 nearest neighbors, they're much more likely to be parallel with their neighbors. So this says there's a microtubule bundle core that is presumably more ancient and polarity sorted a long time ago. And on the edges, you're more likely to be anti-parallel where you can exchange with microtubules that are free in solution. The second consequence is that if the microtubules are interacting with motors that are localized on the edge, the motor is no longer in the center of mass of the microtubule system, which would allow it to induce a torque on the system that it couldn't if it was directly cross-linking the microtubules in the center of mass of the two filaments. What we believe we're seeing is that this induces writhing in the bundles. It's a little tough to see, so I've tried to render it in this plot on the right, where I'm plotting the microtubule center lines. And I hope you can see there's this swirling effect that seems to happen inside of the bundles where the motors on the edge are inducing this twist writhing in the system. Put together, that gives us this overall picture that I think is very different of how people have previously thought about these systems, that it really is steric interactions between adjacent microtubules that organize bundle structure and scale with density, not with direct link to the crosslinker, and that motors navigate this mismatch by predominantly localizing onto the edge of the bundles. So I think taken together, this really brings us towards a multi-scale mechanistic picture of how complex cellular structures assemble that links information about individual motors we can gain from traditional structural biology and cryo-EM crystallography with dynamics that we see at the fluorescent scale. And I think the key thing of doing this is being able to use in cellulo visualization techniques like cellular cryo-electron tomography to visualize interactions of individual molecules. And the sort of final thought that I'd like to leave with is that all of this molecular level imaging is really just the beginning of our problems. And that even if we sort of got to the very end of the structural biology field where we knew where every molecule in the cell was, its orientation, had that link to atomic PDB maps, We then have to figure out the complex rules of interactions of how these things collectively assemble dense cellular environments, which I think is a really exciting challenge that the field is now very well poised to start to tackle. So with that, I'd like to thank everyone who we've collaborated with through the Simons Foundation Network, UC Santa Barbara, who did all of the in vitro reconstitution work, TU Dresden, who have been responsible for most of the plastic reconstructions, and everyone both at NYSBC and the Flatiron Institute who's been instrumental in making projects a success. Thanks.
Thank you, Will. Yo, what a ride. Thank you so much for that very interesting and exciting talk. It just boggled my mind. Anybody got any questions? Okay, Camelia, go ahead.
So, Will, I'm going to preface this by telling you I'm a plant scientist, so you'll figure out where my question is coming, because you have centrioles and centromeres in animals, but you don't or asters in animals, and you don't have this in plant.
Yeah.
Have you thought about how this might work in the assembly for plant cells?
Yeah, I think it's a fascinating question that if your view is really this traditional search and capture thing that everybody nucleates at a pole, you really can't envision how a plant spindle could possibly work. There had been some work out of Arshad Desai's group where they went even further with the centriole depletion and got rid of all the poles, and you can actually still assemble a bipolar spindle and divide. So our sort of view is that the pole is a useful architecture and anchor point, but that dynein itself is probably sufficient to self-organize a microtubule organizing center that can form the same purpose. Though presumably with a centriole-based approach, it's a little more robust to achieving chromosome segregation. But as you point out, plant spindles can do this just fine without poles.
Any further questions? How do you think you could solve the remaining problems, Will?
Yeah, I think one thing that's really missing is something akin to a GFP tag in cellular cryo-ET. Beyond sort of a characteristic like 5 or 6 usual suspects like ribosomes and microtubules, and increasingly like actin nuclear pore complexes, what you often see is just this blob of density that if you wanted to say that's definitely a dynein motor or an egg 5 motor, I think it'd be very hard to convince like reviewer 3 that you were right about that. So Yulia Muhammad's group has started developing these encapsulin-based tags where you could actually directly, akin to a GFP in fluorescent microscopy, mark each individual motor protein. And I think this would just be beyond informative to be able to, execute in cellular environments.
Okay, so we need better tags.
Yeah. GFP resolution, revolution for cryo-EM.
Okay. Any further questions? Well, I think that then is the end of our conference. Would Chart or Lawrence like to make some summarizing remarks? Chart?
Chart too? I'll do it. Chart, go ahead.
Well, I guess well, this has been a great conference in my opinion. Yeah, very large diversity, many different subjects that have been addressed, many different techniques and methods. Yeah, I've enjoyed it really a lot. Great quality of talks. So I would like to thank everyone for your contributions. Yeah, and I'm looking forward to the next Biophysics in Africa conference. I hope that all of you will register again and participate. Yes, thank you very much for everyone's participation. Yeah, Lawrence, do you also like to say a few things?
Yep. My camera. Yeah, I'd like to just echo what Chart said, and I think for sure look at some follow-on activities to organize the community of biophysics in Africa, which is actually worldwide. One of the things that we talked about in this conference and have been talking about for a long time is the Congress for the IUBMB, the International Union of Biochemistry and Molecular Biology, which will be in Cape Town next September. And there'll be biophysics sessions. The the Biophysics Union, IUPAB, will be participating. And I think we'll get some other unions to participate as well. And there is the IUPAP, the physics union has a biophysics committee, council, and it has its 3-year conference, which is this year. And we'll send out some follow-up information about, I think there are some bursary and travel mobility grants available for colleagues that are based in Africa. So I just want to thank everyone for participating in the conference, and look for greater and more things for the rest of the year and into next year. Thanks.
Okay, thank you, Lawrence. I would just like to make a final comment, and I have been enormously impressed by the quality of work that has come from all parts of Africa, and by African scientists in all parts of the world. It's been a remarkable revelation to me to see the very high quality and wonderful work that is being done by so many people. And I think that by organizing an event like this, Lawrence and Chart have done incredibly well to bring us all together. And I think that we should try to stay together and try to work together to advance biophysics in Africa. I think that's all I really wanted to say. And thank you very much for being here and for coming to the meeting. So with
All right,
that,
thanks everyone for coming.